The aim of the following experiment is to use voltage-sensitive dyes to optically record membrane potential changes and synaptic interactions between neurons in the central pattern generators of the crustacean stomatogastric ganglion. This is achieved by loading identified neurons with the dye Di8 and EPQ by iontophoretic injection using sharp microelectrodes and simultaneous extracellular recording of motor nerve activity. As a second step, the fluorescence of the voltage-sensitive dye is excited by green light, which allows the imaging of fluorescence changes that correspond to the changes in the cell membrane potential. Next, the image data is analyzed by selecting a region of interest in the neuron's membrane and comparing the optical signal to the recorded extracellular spike activity in order to identify action potentials and synaptic potentials in the imaged neurons. Results are obtained that show the optical recording of synaptically interacting pattern-generating neurons can supplement traditional recording techniques and open new opportunities for understanding how central pattern generator neural networks function.
The main advantage of this technique over existing methods like extra and intercellular recordings is that after dye filling, optical recordings of membrane potential changes and synaptic interactions can be achieved without the steric restrictions imposed by electrode placement and without further damage to the neurons during long-term recordings.
Though this method can provide additional insight into pattern generation in the crustacean stomatogastric ganglion, it can also be applied to other systems, such as locomotion and respiration in vertebrates.
Generally, individuals new to this method will struggle because dye selection, injection and preparation are difficult, and the parameters for the analysis of the image data have to be set adequately to detect membrane potential changes.
Begin the preparations by making the fluorescent dye solution. In a plastic vial, mix 5 mg of the fluorescent voltage-sensitive dye Di8 and EPQ with 1 ml of F127 pluronic acid in DMSO solution. Next, centrifuge the dissolved dye for 20 minutes at 12,000 rotations per minute in order to separate larger dye particles that may clog the microelectrode. Following centrifugation, use a pipette to aspirate the top 200 microliters of the supernatant. Expel the supernatant into a separate plastic vial. Wrap the vial in aluminium foil to prevent exposure to light. Store the dye solution in a -20°C freezer. On the day of the experiment, defrost the dye solution by placing the wrapped vial in 25°C water for 10 to 15 minutes. To prepare for the recordings, start by isolating the stomatogastric nervous system of a crab and desheathing the stomatogastric ganglion. Apply petroleum jelly around a section of the lateral ventricular nerve, or LVN, to create an electrically isolated compartment. Use modeling clay to attach the petri dish to the operating platform of the imaging microscope. Place a stainless steel electrode wire in the petroleum jelly compartment to record from the LVN. Place another stainless steel electrode in the bath as a reference electrode. When the dye solution is defrosted, draw up enough of the dye solution to fill approximately 2/3 of a microfill needle. Inject the dye solution from the microfill needle into the tip of a glass microelectrode. Cover the dye-filled electrode with a box and wait 15 minutes. Then, backfill the microelectrode with a 3 molar potassium chloride solution. Place the microelectrode back into a dark electrode storage box for 10 to 20 minutes to allow capillary action to pull the dye solution into the fine tip of the electrode. After 10 to 20 minutes, place the microelectrode into an electrode holder and attach it to the head stage of the intracellular amplifier. The intracellular amplifier is connected to the data acquisition board that provides a pulse signal to drive current pulses into the microelectrode. Next, adjust the microelectrode to an oblique angle and position its tip above the stomatogastric ganglion. Lower the microscope objective until the electrode tip is in focus. Then, lower the objective, being careful to stop before touching the microelectrode. Then, lower the microelectrode until it reappears in the focus of the objective. Monitoring progress on the oscilloscope, gently pierce the membrane of the selected stomatogastric ganglion neuron. At this point, compare intracellular and extracellular recorded action potentials in order to identify the impaled neuron. Begin dye injection by switching the intracellular amplifier into current injection mode and injecting 10 nanoamp positive current steps into the microelectrode. The current step should last for 1 second, separated by 1 second gaps of no current. Continue this current injection for 30 minutes. After 10 minutes of dye injection, use the MyCam02 imaging system to check the dye spread within the neuron. Repeat this check after 30 minutes. If the dye filling is successful, the dyed patch around the microelectrode should grow between the 10-minute and 30-minute images. Pull the electrode out of the neuron and allow the neuron to relax for at least 20 to 30 minutes to allow the dye to spread further in the cell. During this time, another neuron can be injected with dye. Switch to the high light transmission efficiency 20 times objective on the MyCam02 imaging system. Reposition the microscope's operating stage as needed and focus on the dye-filled neuron. Feed the extracellular recording data into the imaging system through its analog input channel. Turn off the room lighting and all remaining light sources. In addition, close a black curtain around the recording rig to prevent light exposure from external sources. Set the imaging system to the appropriate pixel resolution and imaging time shown in the chart. If the dye in the neurons is not very strong, use the H-bin setting. Set the microscope's light level as low as possible to avoid photomodulation and photo damage of the recorded neurons. Set the imaging sessions to between 4 to 16 seconds long. Then, record the spontaneous activity of the dyed neurons over several imaging sessions. To begin analysis of the optical responses using the BV-ANA imaging software, select an appropriate circular region on each recorded neuron. If temporal smoothing of the data is necessary, for example, if the dye generates a relatively low amount of fluorescence, use the software to apply temporal smoothing with 3 to 5 time units. The two dye-injected neurons in the upper left of this figure are identified as a PD neuron and an LP neuron on the basis of their intracellular recordings. The voltage traces to their right show the spontaneous optical responses from each neuron. The extracellular recording of the LVN is shown at the bottom of the figure. The LP and PD neurons are part of the pyloric central pattern generator of a crab stomatogastric ganglion and are mutually inhibitory. Their optical recordings can be matched well to the extracellular recordings of the LVN. The inset at the bottom shows an overlay of several sweeps of LP action potentials and their average, demonstrating the match between optically and electrically recorded activities.
Once mastered, this technique can be done in 1.5 hours if it is performed properly.
Following this procedure, other methods like immunostaining can be performed in order to answer additional questions such as which type of synapses was involved in eliciting the observed membrane potential changes.
After watching this video, you should have a good understanding of how to record optically multiple identified and synaptically interacting neurons in the stomatogastric ganglion.